proteome itself, I already told you about a mechanism called as ERAD, endoplasmic reticulum associated degradation. If there is some protein is still stagnating there means, then the endoplasmic reticulum is having a mechanism to degrade the protein completely. Like that, in transposition, that translocation also, you are bringing a very good point about that thing. Actually, it's having a mechanism. Only in certain cases, those mechanism is not working. How it is escaping, we need to find it out. Especially in the case of the BCR-ABL tyrosine kinase, CML, chronic myeloid leukemia, it is a pretty good thing. We need to find it out. I am trying it, but I can't find it out. You are most welcome to answer for that.
Yes, sir. So I think
I'll answer this call and come back to you.
Okay, sir.
Yes, sir, you can tell. So I was working on that. I was not able to find it out. I am having a lot of articles related to that. But there are some interesting articles related to that, what is that, lymphoid leukemia. What is that, ALL, acute lymphoid leukemia, which is mainly the problem with the kids. They have done a lot of work on that, and now they are able to cure it. ALL, yeah, acute lymphoid leukemia. Yes, I am having articles related to that. From that only, I used to say that we are looking at the final stages. That means that after translocating the Philadelphia chromosome, we are targeting only the BCR-ABL tyrosine kinase. But we need to go one step behind that and find out how it is translocating. If it is translocating means, then how the chromosomes are recognizing this and they are coming and picking it up. That is the bigger question. So I am trying to find the answer for that. But we'll try it. Yes, Adar, now you can answer.
Yes, sir. What I'm thinking is like the lymphoid or myeloid cells, and it mainly happens in the bone marrow. So the cells over there are pluripotent.
Yes.
So does the pluripotency plays a role in it?
Yeah, that's a bigger question, because initially it starts and finally it multiplies like anything else. So we need to find out how it starts, because nature itself is having a repairing mechanism. If it is doing that, immediately it will come and fix it. But it is not happening in certain type of cases. That's what we need to find it out. So only they are doing it with the bone marrow transplantation is mainly for CML. They are using bone marrow. But if you look into that bone marrow regeneration, it is not that fast. When comparing with the growth of CML, the chronic myeloid leukemia, even though if you are doing that bone marrow transplant and doing that thing, the progression of CML is too much when comparing with the bone marrow transplanted thing. Yes.
Yes, sir. Means that is because the defense system, like the macrophages are only like means those are the faulty ones. So
Yeah,
that's why there are no other cells to take care of it.
Yes. Actually, it's very fast. That's what happening in the cancer cases. If you want to do something, even overnight itself, the progression is very fast. We can't even think about that. The growth of the cancer cells are very fast. So even though we are are doing transplant or whatever, the bone marrow transplant and everything, it is escaping from a lot more things. We can't even imagine those things. That is the problem we are facing. Even though our technology and everything is developing, but for most of the disease, we are nowhere to find a cure. That's what, unless or otherwise, if we are not able to find out the mechanism, we can't do anything. Another problem with these cells, whatever the cancer cells is, they are all highly glycosylated. So if they are all highly glycosylated, radiation will never work on those cells. Because they are giving both chemo and radiation therapy for most of the cancer. If you are giving radiation therapy, only normal cells are dying, not the cancer cells. So we need to find out a way for this, and then you are taking almost all the chemo and radiation means your immune system will go down. Because the natural immunity, it will try, it need to boost up. So whatever, that is the main thing, whatever we are doing now. So even though the foreign pathogens are infecting us, whatever the vaccine we are taking, it's actually enhancing our immune system to make them prepare for the foreign pathogen. That's what's happening. So we need to find out a way to enhance the immune system. But we are not doing that in cancer. Because our immune system is completely going down. Yes, others.
Yes, sir. Yes, I got it.
Yeah, because if you look into the viral pathogen, what we are having now, actually we are helping try to improve or enhance our immune system. But in the case of cancer, our immune system is actually going down. So we need to find out a lot more way for that. So whatever the therapy we are having, there are a lot more works are going on. So that's a bigger thing. We need to find it out. So if you look into the history, we are talking about tobacco and all the other products. Some of the exceptional characters are there. If you look into Winston Churchill and Fidel Castro, they never had cancer. They lived a very long and healthier life. Only certain population is prone for this when they are consuming the tobacco-related products. They are coming up with cancer. And some of the kids, especially in the case of chronic myeloid leukemia and ALL, they are not at all exposed to any of these tobacco products. But what is the reason behind that? Why they are getting those type of cancers? That is another bigger question. We need to find an answer for this. Because they are not exposed to any of the habits. Why they need to get that type of diseases? Both the case of acute lymphoid leukemia and CML. Some of the cases of CML are just they will diagnose it after taking all this medication, they will be all right. So we need to study the system properly. So that's what I already told you, what is the bottleneck we are having in our country. The clinicians are far, far away from the researchers. So we are not having a very good relationship between the clinical person and the researchers. But if you look into almost all the Western countries, they will be located in the same place. Most of the doctors, after finishing their masters, MBA and everything, they will all do PhD in US. MD, PhD PhD. Most of the doctors will be MD, PhD. They will do the clinical thing and also do research also. But in our country, there is a gap between these two things. So once the gap is minimized, I'm not telling that it need to be minimized, that's coming close together, then we can achieve a lot more things. And not only this, our government is spending a lot more money on health sectors. We can reduce those things also. So that's what we need to take it to the next side. Bioinformatics, you are all very good scientists, you know what's happening. So based on that only we need to find out a solution for that. The translation research is very important. Whatever you are doing in the lab, it needs to go to the bed immediately. So that is very important thing. So unless or otherwise, if you do not know the mechanism, how the disease starts and how it is progressing, so there is a big bottleneck with this corona also, COVID-19. Why? Because each and everyone is telling one day one inhibitors and other inhibitors. But whatever they are telling, you can't do anything based on that. You can't design drug for that because that protein is playing a very vital role for other functions of the body. So if you are blocking that, that's what I'm telling. ACE2 inhibitors, they are telling. Angiotensin converting enzyme. Because spike protein is binding to that. If you are blocking it, it's having a lot more other functions to do for us. Then your lungs will become plastic. So we need to thoroughly study the virus, how it is escaping our system, what is the mechanism it is using. So for that only we are having different tools and everything. We need to implement it to find out a cure. That's what's happening everywhere. A lot of people are working to find out a suitable drug candidate for this. It's still going on. We'll try to find it out. Okay, so I'm not sure whether I answered your question or not.
Yes, sir, I got it.
No, we are there. We need to find it out. But that is the case. Whatever I told you, no, it's translocating. At that time itself, it's having a mechanism to block it. But some cases it is escaping that, and finally we are coming up with this type of diseases. Some of them are age related. That if the age is above 75 and everything, they will accumulate with certain type of diseases. So those are all age related. Once it is age related means then it is relating to our biological system, because the biological clocks are there where it will switch it off. So that is another bigger question we need to answer it. So let's find out whether protein-protein interaction is going to play a vital role or not. So yes.
Sir, I had one more question.
Yes.
So, is it similar to means as the transposons are not very useful, like they disturb the system. So, does aneuploidy happen in mitotic cells?
Yeah, there only it starts separating. Once it starts separating only, everything is doing that. It is that particular chromosome. I'm having that number. I don't want to tell the wrong number. I'm having it in my article. So while doing that only, it is translocated and it is going, and whatever it's going to express is the BCR-ABL. Whatever BCR-ABL structure we are having is smaller part. It is a very big protein. So that's what we need to find it out, how it is, because if it is if it is not in the normal form, then our system need to abolish that thing. But if it is not abolishing means that translocation is accepting means then there is something is happening. That's what I am telling you from day one onwards. This machinery, whatever I am telling you, it's 99.9% foolproof. It is having all the checking mechanism. Once something goes wrong, it will immediately shut it off. It's a very good system. Even the PCR, whatever we are using, the error is 1 in 1 million. That is the same thing happening in our system also. The ribosome machinery will never make any error. If it is making error, that error is 1 in 1 million. So the error is very less. But sometimes some things are happening that we need to find it out, how it is happening, because something is abnormal from the nature one means, then it will try to stop it or shut it down. If it is not doing that, then there is some other mechanism got triggered. So that's what we need to find it out. We need to do a lot more research on that. Oh, sure, I'm pretty sure we will come up with a lot more solutions. Yes, sir.
Yes, sir. I got it.
No, that's what I'm having the same question. How it is progressing. Once this translocation is taking place, because it is moving from a different chromosome, and that chromosome is accepting this means, how it is accepting, especially in the case of Philadelphia chromosome. How it is happening, how it is. It's not so easy to answer. When I started it gives me a lot more question, because we are starting from that point only, where BCR-ABL comes. From there, because they already identified this as a target, and they are progressing from BCR-ABL only. There are very less data when you go how it is happening. It's not that much. It started with there. It is translocating. From there only it started. How it is started, no one knows. They are all working. They are giving a lot more reasons and everything. But we are the one needs to find it out. Everything is there. We try to find it out. Okay?
Yes, sir.
Okay. Yes, Somnath. No question from anyone else, then I will go to the PPT. Okay? So, protein-protein interaction, biological aspect of this. So, since me and you are having a common language, I am telling you whatever I want so that you are fulfilling that thing. So, but if you look into the cells, how they are communicating, so that is very important. So, the importance of the interaction playing a very vital role. There are different types of protein interaction, and what are the methods to identify this thing? These are the main things we are going to discuss here. So, the main thing is proteins are the one which control and mediate many of the biological functions or activities of the cell. If you look into the cell, a cell is not static. It changes in shape, and then division, cell division will take place, and then metabolism. And then if you look into the cells, all the cells are not equivalent. Each and every one is having a different function and are not equivalent. Even if you look into the protein, which is there in the liver, it may have the sequence homology, but there may be some differences in some part of the protein sequence. It may be the same protein. It is coding in different places. It may also have the same function, but there may be small difference will be there. So why it is making this difference, that we need to study. So based on the similarity only, we are using BLAST and FASTA to find out the sequence similarity, to find out the protein structure and everything. But if you look into the place where it locates, why it need to have that much sequence differences? So each and everything we need to find it out. So the another thing is why protein-protein interactions are so important. So what will happen if a wrong protein is coming and binding to a different protein, then the functionality of the protein will be entirely different. If it is a transporter, means it needs to transport something. If it is not having the message from that thing, then it is going to do some malfunction. So there are a lot of things are happening. So, and then each and everything needs to do the function of the thing. That's what happens even if you take the simpler thing like glycolysis. Everything needs to be do it in a proper way. If the production of H plus ion is too much, then automatically the ATP synthase will start activating it. And then it needs to produce ATP. If it is not producing ATP, then there is a lot of malfunction will take place. So that's what happening. There is a type of disease called as LHON. LHON is the name of the disease, which is called as Leber hereditary optical neuropathy. The eye of the person may be good, and the brain is also good, but the problem is the pathfinding nerves. It's having some defect. The nerve which is connecting and which is carrying the message from the brain to the eye, if there is some malfunction is happening, then it can't send the message. So finally, the person will have the eye problem. He can't see any smaller objects. But you can see the eye is looking good and the brain is also looking good. It is the problem with the pathfinding nerves, or otherwise called as axon regeneration. The regeneration will never take place. If you look into some of the organisms like frog or otherwise goldfish, if you cut those pathfinding nerves, both frogs and these goldfish, this axon will regenerate and it will recapture the brain and it will reconnect the brain and the eye. It will carry the message. But in the case of human beings, the axon regeneration will never take place. The pathfinding nerves, if there is some problem, it will not regenerate. So, but it is a rare disease, 1 in 1 lakh, it will be there. But there are a lot of diseases where we need to find it out. And this LHON is mainly happening because of the mutation in the mitochondrial DNA. Only one point mutation, I think, 1 or 2 amino acid gets mutated in the complexes. Mitochondrial complex. Finally, because of that mutation, the pathfinding cells are not taking the message to the brain. So the person looking like a normal person, but his eye vision will drop down to 60% or 40%. It will happen within a week, between the age of 10 to 20. Suddenly that boy or a girl, most of the time these diseases are happening to boys. The percentage is very less for girls. Most of them it's targeting only the boys. So finally he will lose the vision. So this is one of the type of mutation happening based on our topic, protein-protein interaction. If the messages are not transporting, then it is a disorder. So only we need to spend a lot more time to study what type of mechanism and how it is happening. Okay? If you have any question, we will discuss. Hello? Hello?
Yes, sir.
If there is no question, then we will continue in the next class. Is it okay?
Okay, sir.
Okay, thank you. Take care. Bye.
Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you, sir.
Thank you, sir.
Thank you, sir.